So my name is Andrew Kodani. I work at St. Jude Children's Hospital. Originally, I was at Boston Children's Hospital with Emmanuel Gassoni and Dr. Christopher Walsh. And this is our first foray into epigenetic disorders. And so I want to just start by introducing how we got involved with Kabuki Syndrome Foundation. And so this was a family friend of Leila Zenia who put us in contact with Leila, that then allowed us to work with Janet Lee, who then introduced us to Jackie Harris and Brittany Simpson, who then helped us obtain patient samples and to get the project started. That then led to conversations with Dr. Hans Bjornson and a continued collaboration with my long-term mentor, Dr. Emmanuel Gassoni. Through the findings, we were able to recruit multiple members of the St. Jude Children's Hospital who are involved in developing useful therapeutics for delivery into children. So we were put into contact with multiple members of the St. Jude community who have developed useful therapeutics for multiple childhood disorders, including Dr. Richard Finkel, who came up with the ASO therapeutic that has now treated SMA, which is early childhood disorder that leads to early death. And so we then put in contact with Dr. Paul Taylor and Dr. Asim Ansari, who are helping us. And so we're working in collaboration with 6 biotech companies in order to develop a useful therapeutic for Kabuki Syndrome Foundation. And so where we start is the advent of gene discovery, which was really skyrocketed by the advent of molecular genetics or next generation sequencing that led to the discovery of a number of disease causative genes. But what has lagged behind is the biological understanding and the development of useful therapeutics. And so there's this lag time where we need to have the biological understanding and the therapeutics catch up to the gene discovery. And so because of that, there are only 3 ongoing trials. And so we need a better molecular understanding of Kabuki syndrome along with other childhood disorders. And so my lab is focused on trying to understand the molecular basis of all these childhood disorders. Therefore, we can create targeted therapeutics that are useful in clinic. And so we work with physicians and geneticists from around the world, including Dr. Harris and Dr. Simpson, in order to identify individuals who have certain diseases such as Kabuki syndrome. We then utilize mouse models similar to Hans Bjornson, and we develop stem cell models to mimic the human disorder. And then we use mechanistic studies, including high resolution microscopy, biochemistry, and machine learning in order to understand the mechanistic sequelae of various complex disorders, including Kabuki syndrome. And so as Alessio very well described, DNA is normally repressed in this nucleosome complex, which helps to suppress the DNA from being open and active, but comes along these complexes of enzymatic proteins, including KMT2D and KDM6A, which help to de-repress the DNA, allowing for transcription factors in order to turn on certain genes. But then the question then becomes, which I think all the people in this section have, which is what genes turn on and off? As Max showed, thousands of genes get turned on and off within C. elegans. And so in order to create a novel model to study Kabuki syndrome, we collaborated with Dr. Chandra Miller at St. Jude in order to develop new knockout systems for KDM6A and KMT2D using CRISPR-Cas9. And using this technology, we were able to develop 2 cell lines each of both KDM6A and KMT2D knockouts. What we found, which was a novel finding within the Kabuki field, is in this Western blot, you can see that KMT2D is absent from the KMT2D knockouts, but is also reduced in the absence of KDM6A. And similarly, KDM6A is reduced in the absence of KMT2D, and it's absent in the KDM6A knockouts. This suggests that these 2 proteins function together in order to promote chromosome opening or closing, depending on which area it's repressing or activating. And so in order to determine what genes get turned on and off, we use KDM6A knockout immortalized cells, mouse embryonic fibroblasts, and human neural precursor cells in order to understand which genes get changed using RNA-seq. And this data was collected and analyzed with the help of Dr. Michael Wong here at St. Jude. Similar to the findings of Dr. Hans Bjornson at VAX, we ended up detecting over 5,199 permutations of changes within the transcriptome of human cells. And this is an astronomical number that is not testable in a realistic setting. And so we start to do what's called gene clustering or functional analysis. And when we start to cluster all these genes in order to see how they commonly function to promote cellular function, we ended up focusing on this class of genes that control the formation of a structure in the cell called the primary cilia. And we found that 67 out of the 289 annotated genes actually are involved in the formation of primary cilia. We then used a higher level algorithm looking at disease association and function. And then we settled on 13 cilia essential genes that, when mutated, cause human disease. And so when we start to look at Kabuki syndrome, as has been well described by everybody previous, we see that there's clinical manifestations such as a cleft palate, liver cysts, kidney malformations, that then are recapitulated in a class of diseases called ciliopathies. And so we see the common phenotypes within the ciliopathy patients that we see in Kabuki syndrome. So the question is the gene changes related to a change in cilia within Kabuki syndrome individuals that then manifest in human disease? And so to test this, we want to first see if cilia themselves are affected. And this work was done by my talented grad student, Taylor Hedgecock. And you can see the primary cilia as a hair-like structure seen here in green, which is surrounded by a ciliary membrane. And in the knockout cells, compared to the wild type, you see a nice long elongated cilia. However, in the KDM6A and KMT2D knockout cells, and within the stem cells, we find that cilia are shorter, but they're also malcomposed, as you can see a reduction in overall RL13B or ciliary membrane markings. And this was quantified to show that there's a reduction in ciliary length. And this is not a trivial task, as Taylor painstakingly looked at a microscope and measured the length of hundreds of cilia per experiment. And he found that normal cilia are not present in the KDM6A and KMT2D knockouts, suggesting that cilia are abnormal. But ultimately, we need to know, how are they abnormal and how do we fix this? And so primary cilia themselves are hair-like organelles that function to transduce growth signaling. And so you have a ligand here as a hedgehog in order to display the pathway that's involved in growth signaling called sonic hedgehog, that then goes to the primary cilia, activates, and then leads to a signal transduction cascade that ultimately leads to increased growth signaling. And when you don't have a primary cilia or it's abnormal, even though you have the signal, it cannot transduce. Therefore, you get a reduction in growth signalings that ultimately manifests in the malformation of a number of different tissues within the body, thus classifying it as a ciliopathy. So then we wanted to test whether the cilia within the KDM6A and KMT2D knockouts can signal. And when we have this micrograph that is shifted, denoted by the arrow, we see that the ciliary membrane seen here in green, when treated with the driver or the inert chemical that the drug is dissolved in, we see that there's no activation of the receptor smooth end, which localizes to the primary cilia. However, when we treat with sonic agonist, which mimics the addition of sonic hedgehog, we see that in the wild type cells, we see the presence of smooth end within the cilia. However, the cilia within the KMT2D knockouts and the KDM6D knockouts fail to translocate smooth end into the cilia, therefore they are inactive. So can we restore cilia function as well as ciliary composition within these knockouts? And so Taylor then started to look at those 13 different genes that were disrupted that are involved in ciliary formation and found that the only protein that was affected on the immunofluorescent level was this ciliary repressor called CEP97. And when we look by Western blot analysis, you see that there's a significant increase in the stability of CEP97, which serves to sit on top of the primary cilia and repress its formation. So the ultimate question is, if we were to deplete the cells of CEP97, can we restore primary cilia length and composition? And so this work was done by my postdoc, Emilia Asante, as well as Taylor Hedgecock, and where we use ASO, which is an antisense oligo that helps to deplete the expression of certain genes. We find that by treatment with the control ASO, we see no change within the ciliary length because it's an inert control. Whereas when we treat with the CEP97 directed ASO, we see that on the left, you can see that there's a reduction in the amount of CEP97 down to the levels of control. But when we look at the primary cilia itself, the wild type is shortened, as well as the KMT2D and KDM6A knockouts are still short, suggesting that though the levels of this one ciliary repressor is increased, reducing the levels still does not correct the defects associated with the cilia within these knockout cells. And so we went back and looked at the other 13 genes, but then we also loosened the stringency to look at genes that are not disrupted by multiple fold, but looking at disruptions of less than one fold, which may have catastrophic effects on the formation of primary cilia. And due to non-disclosure agreements, we can't reveal the name of this gene, but protein X localizes along the length of the primary cilia. And you can see this in wild type cells. However, in the absence of KDM6A and KMT2D, it is completely absent from the primary cilia. And looking at the protein levels, we find that this protein is absent from all of the knockouts, suggesting that disruption of the expression of protein X might be driving the ciliary phenotype in these knockout cells. And so because we have a loss of the expression of this one factor required for primary cilia formation, we said, if we add this back, can we restore cilia length and can we restore cilia function? And that's exactly what we see, but let me walk you through it. So in wild type cells, we have a normally long cilia. However, in the KMT2D or KDM6A knockouts, we have very short cilia, and we use GFP as a control, and that was well described by Max Hyman. And so we then expressed a GFP form of protein X. And we found that just by expression of protein X alone, we can restore primary cilia length in the KMT2D and the KDM6A knockouts. And I don't have the time to show you today, but the overall function, as in terms of the signaling, can be restored upon expression of GFP protein X. And so to summarize the things that I found is that loss of KDM6A and KMT2D, it was surprising to find that they're required for the formation of normal primary cilia, and decreasing the level or expression of protein X then leads to the formation of defective cilia. And so the roadmap of where we're going now is that we're starting to develop adenoviral therapies in order to reintroduce protein X to then use observations from the clinic in order to see whether it's efficacious. And more importantly, in a targeted therapeutic domain, we are then partnering with partners within the biotechnology field in order to assess whether ASO therapies to increase the expression of protein X is a viable target using antisense oligos. And going forward, we're in the process of starting to collect patient samples from Dr. Harris and Dr. Simpson in order to understand the mechanistic sequelae of Kabuki syndrome from the perspective of patient tissues. And in that process, we're going to create a biobank here at St. Jude, which will enable other researchers to use these valuable primary tissues that will be available through St. Jude, with the hopes of eventually creating a center for excellence at St. Jude to enable people that are affected by Kabuki from around the South in order to receive assessment and treatment at St. Jude. And ultimately, we'll continue our functional studies and collaborations with Dr. Hans Bjornson and Dr. Emanuela Gassoni to test whether the AAV therapies and the ASO therapies are efficacious within mouse models. And with that, I really want to thank all the people in my lab who have pitched in to this amazing opportunity to work with the Kabuki Syndrome Foundation. These are the people in my lab. These are my senior research associates, and this is an ambitious high school student who has made great strides in understanding the disease from like a machine learning perspective. And with that, I want to thank you guys for the opportunity. Thank you.